Hi everyone, this is Solutions for Microbial Genetics webinar. If we could just give it another minute or so, because we've got a lot of people still joining. Bear with us. Welcome to the latest Novogene webinar, Solutions for Microbial Genomics. I'm John Cafferty, Marketing Manager here at Novogene Europe. This is the final webinar in our lockdown series. We started the series in April to provide an online alternative to all the conferences that have been cancelled. We've had almost 2,500 people registering for the webinars over the course of the series. We'd like to thank you all for being part of this. We'll be taking a short break over the summer and we'll return in September with a new programme of webinars. In October, we have the Novogene European User Forum. This will be an online event with guest speakers from industry and academia. We'll be publishing both the webinar and the forum programme soon. A bit of housekeeping. The webinar will last 1 hour with a 10-minute question and answer session at the end. Please submit your questions via the question and answer chat functionality you'll see in the bottom of your screen. We'll answer as many questions as we can, and if we can't answer your questions live, we'll get back to you by email after the webinar. I'd like to introduce your host for today, Gürkan Korkmaz. Gürkan did his studies in biotechnology in Germany and Sweden. His research focus at Uppsala University covered bacterial protein synthesis and codon usage bias. After his academic path, he moved to Cambridge as an account manager at Illumina and joined Novogene in April 2019. Gürkan leads the DAC team at Novogene. Gürkan, over to you.
Thank you very much, John, for the kind introduction. Yes, my name is Gürkan Korkmaz, and I'm the sales manager of Germany, Austria, and Switzerland, which is DACH. So before we start today's webinar, I'd like to introduce you the company a little bit for you to get a better overview of who we are and what we do. Novogene is a young company. We were founded in 2011 and we are headquartered in Beijing. Since day one, Novogene has been dedicated in advancing genomics and improving life. We do this by using cutting-edge genomics technologies and high-performance computing. Our customers are profiting from the access to the most recent instrumentation and methodology in the genomics market. Novogene has one of the largest sequencing centers in the world. The combined output of all our NGS instruments would result in more than 280,000 human whole genome sequences. Or in other terms, that would translate into 25 million microbial genomes. Our sequencing centers and offices are distributed throughout the world. Our largest sequencing centers are in Tianjin, just outside of Beijing. We also have subsidiaries in Hong Kong. We have a lab in Singapore. And our first overseas laboratory opened in 2016 in California on the campus of UC Davis. A few years after that, in 2018, we have opened our first lab in Europe, where we are serving our European customers with a lab and offices in Cambridge. And also most recently, this year, we are happy to announce that we also have a legal entity in the Netherlands. This slide gives you an overview of what sort of genomic solutions Novogene can support you with. The range is quite large. We do quite a few projects on plant and animal genome sequencing. We also do regulatory aspects of the genome with epigenetics, as well as, of course, transcriptomic solutions. Noteworthy, of course, for today's topic is that we also offer RNA sequencing of prokaryotic samples. Today in our webinar, we are focusing on the services regarding the microbial genome. Here we can offer de novo services where no reference genome is available. Here we would use usually a combination of short and long read technologies. We do offer, of course, resequencing of your samples where we do have a reference genome, for example, for the detection of SNPs. We're also heavily involved in the area of metagenomics from various environmental samples based on shotgun metagenomics or amplicon-based metagenomics. So I've showed you on the previous slides that Novogene is quite a large company with thousands of samples that we receive each and every day. We have completed thousands of projects to date. One project that we cite often is a metagenome project where we supported the University Medical Center Groningen in the Netherlands with a sequencing of 10,000 samples. We are very proud of such collaboration. It is also important for us that we are equally dedicated for smaller size projects. These big numbers might be intimidating for some, but the smallest project size that we have that we accept is 1 sample and 1 gigabase. So if you have small projects, do not hesitate to contact us. Data quality and the dedication of our service is identical with our large-scale projects. We're quite a big team in Europe. We have more than 25 representatives in the field across Europe that you can reach out to and discuss your project in detail. In most cases, we can also support you in discussing your project in your local language if that is something you prefer. So please don't hesitate to contact us. The easiest way is to write an email to europe@novogene.com. From there, your email will be directed to your local contact person. You can also contact and connect with us through our website, Twitter, and LinkedIn. So that is so far from me. And now I'd like to introduce you our speaker of today, that is Dr. Wei Li. Wei got her master's and her PhD from the Clemson University in the United States, where she has been working on the cotton transcriptome. She started her career at Novogene in the bioinformatics department as a senior bioinformatics engineer. In August last year, she was promoted as our senior product manager. That's it from me. Okay, Wei, now it's on to you.
Thank you for the introduction, Gökhan. So I'm going to share my screen. So can you all see my screen?
Yes, looks great.
Okay. So again, thank you for joining us for the webinar. And today I will give you an introduction about the solutions for microbial genomics provided by Novogene. So in my presentation, there will be 3 parts. First, I will give you an overview about our microbial services. And then we move on to the details for each major service. And in the last part, I will give you some case studies information. So first, I'd like to start with my presentation by showing you this agar plate. So this is cultured by Tasha Stern. She is a lab technician in United States and also the mother of an 8-year-old boy. So she cultured those microbes after his son placing his hand on this agar and he was playing outside, and then he came back and placed his hand on this agar. So it seems like all those microbes are surrounding us every day. So those microbes can change the soil condition to modify the plant growth, or they can also serve us some like wines or cheese, and they also cause disease. Or maybe they will clean up the water and change the environmental conditions. So that's how close the microbes are to us. How close we are to the microbial world. So actually, based on the publication in 2016, on Earth, there will be almost 1 trillion microbial species on the Earth. However, we only sequence very small proportion of them. So only 10,000 of microbial species has been sequenced. And even smaller proportion, 10,000 of them can be cultured in a lab. As we all know, so the cultivation on agar will be a very common way or fundamental method to study the microbes. However, what if we cannot culture them in the lab? So what is the most effective way to study them? Well, in Novogene, we provide you both solutions. So if you culture your microbials, if you culture the microbes on agar, so you can send us your single or individual species for de novo sequencing or resequencing. If you don't culture them or you fail to culture them in the lab, so you can send us the community, the whole community, by using the amplicon-based metagenomics or shotgun metagenomics to study the community. And we also have the solutions or services for the RNA-seq of the microbes. So you can sequence the single string as well, and also study the community by using the Meta-transcriptomics. However, in today, we will only focus on those genomic sequencing services, the 4 listed on the top. So in this table, I listed the major services we provided for the microbial projects. For different services listed in the first column, we will provide you solutions or options by using different platforms, such as the Illumina platform by using the short resequencing method, or the PacBio and Nanopore for the long resequencing method. And for each of those services, they will meet your different research objectives, such as Amplicon Sequencing and metagenomics, will help you to discover the diversity in your community. While for the single-strand sequencing, you will study either the genome structure or to check out the variation in the specific culture. And for the RNA-seq, for the single species, we will focus on the transcriptome quantification and the structure prediction. So this is the whole map or the overview of our services. Now let's move on to the details for each of the service. First, we will start with the DNA sequencing for the individual species. That will be de novo and resequencing. If you don't have any idea about what it should look like, then you need to choose the de novo sequencing because there is no reference genome available in our database. And we have to perform the de novo assembling by putting those pieces together one by one. However, if you already have the reference genome published, then you can choose the resequencing. So in this way, we can detect the variation in your specific culture. Well, for different services, we have different sample requirements. For the de novo services, it depends on which platform you choose to use. For the Illumina, we will provide the short read sequencing and PacBio for the long read sequencing. So why we need to sequence long reads? That really depends on the genome structure of your species. If there are a lot of repeats or heterozygous regions in the genomic DNA, so the short reads assembly will not overcome or avoid some low accuracy regions for those repeats and heterozygous regions. While the long reads will perfectly get across those regions during the assembling. So with the long read data, then we can perform better assembling for your species. And also for the Illumina sequencing, since we include the PCR amplification during the sequencing process, it will also introduce some bias. While in the long read sequencing, we will skip or there will be no PCR amplification in the sequencing part. So that also avoids some amplification bias in the sequencing part. So it really depends on which strategy you prefer to proceed. And we provide different data deliveries as listed here. By using only NGS data from Illumina, we can only construct the draft map for both bacteria and fungi. Because for those, we cannot assemble them very well as there may be some repeat regions and heterozygous regions, as I mentioned. Well, if you choose the PacBio sequencing method, with those long read data, we can finish the complete map for bacteria and also the fine map for the fungi. So it really depends on the genome size of your fungus. So if you got larger size, then the contic size will be different as well. Well, for the bacteria, we will deliver a scaffold without any gaps in the reference genome. So those different strategies will help us to explore the genetic structure and some functions or even evolutionary information of your species of interest. To perform the analysis, we just listed the general steps for both de novo and resequencing. For the de novo, again, our major point is to check out the genomic components. And we will perform the de novo assembling and then annotate those components with the biological functions. For the for the resequencing part, we will map all those clean data against the reference genome, and through that step, we can detect or call out all the variations in your specific culture. So for different projects, we will deliver different results. For the de novo, here I showed a complete map of a bacteria. So in this part, you can see the perfect scaffold outside, and also we will label out the coding genes, the gene functions annotated by using different databases, and also some non-coding RNA information and the GC information in the center. Well, for the resequencing, as we focus on the variations, so outside again will be our reference genome or chromosome, and then we will include the information for SNP, indel, and the copy number variations, and also structural variations in the center. Well, that will be everything for the individual species. For the microbial community, we will have two choices as well. So Amplicon sequencing and also shotgun meta-genomic sequencing. So what is Amplicon sequencing? This is a very cost-effective way to study the microbial community. we usually amplify a specific DNA region of the marker gene. And to target different microbial community, we will use different marker genes, such as for bacteria, we will amplify the DNA region, which will encode the 16S ribosome RNA. And in each of those marker gene, we will see both highly conservative region and also highly variable region. By comparing the sequence similarity across samples or across the PCR products, we can perform the species annotation, and that will give us the information about your community. For the Amplicon sequencing workflow, we will start with the sample preparation and then sample QC. And we will also perform the PCR amplification, library preparation, library QC, sequencing, data QC, and bioinformatic analysis. So on the right, I listed the pipeline for the bioinformatic analysis for the amplicon-based metagenomics. So here we will use the OTU clustering after we get the clean data. So what is OTU? OTU is an operational taxonomic unit, has been defined by us. So you can choose the cutoff for the similarity to perform the OTU clustering. And after getting the OTU, we will annotate them with their taxonomic information. And with that information, you can perform both alpha diversity or beta diversity to check the community differences. And we also include or provide the statistical analysis as well. So here are more details about the OTU clustering. So during our amplification, each of those kind of small bar will be one PCR product. And in the middle will be our target region of our marker gene. On both ends will be the barcodes we have been added or included in the PCR process. So with those barcodes, we can distinguish those PCR products from different samples. So we call this step as demultiplexing. However, is the barcode always necessary? Well, if you go to a glossary store, so you will use the barcode to check out or distinguish different foods or different goods. Well, this is also being applied in the PCR process of our amplicon sequencing. We use an 8-base sequence in the PCR products to distinguish or to label each of our samples. So during this process, we amplify the 16S region, and then we add a barcode, and then we move on with the library preparation. So again, with this information, we can successfully demultiplex samples. This barcode is not always necessary, but we highly recommend you to include the barcodes. Here are the reasons. First, if you have the barcode attached, then you can pull multiple PCR products from multiple samples within one library. If there is no barcode, we have to construct one library for each sample. So we can accept both genomic DNA and PCR products from our clients. If you send out the genomic DNA, we will definitely include the barcodes during our amplification and library preparation. However, if we receive the PCR products, if there is no barcode, we will request more input products from you because we have to construct one library for each of your sample. So that makes everything easier if you can add the barcode during your PCR process. So after the demultiplexing step, then we will move on with the OTU clustering step. In this part, as I said, within each sample, we will annotate each of those OTUs with the taxonomic information. And after that, we will get the diversity study and also the abundance analysis for you. So for our OTU clustering, we will use the 97% as the cutoff for the sequence similarity. That means anything or any sequence with the similarity higher than 97% will be clustered together as one OTU. So here I'm showing you the results from the OTU clustering and species annotation. So the bar graph is kind of showing the OTU counts from each samples. And also you can check from this video. So we also provide an interactive page to show you the information of your samples. You can change the counts for the OTU and then refresh this table. Then we will list the annotated OTUs in all of your samples. With those annotation information, we can move on with the diversity study. So we will include both alpha diversity and beta diversity in the analysis. Alpha diversity is to estimate diversity within a sample or a set of replicates. That means a group. So with this information, you can go ahead to explore the species richness in your community by using the OTU counts, or you can study the species diversity by using the alpha indices as shown on the right table. Well, in the beta diversity, we are going to estimate the community dissimilarity. You can use two different informations. You can use the unweighted information or annotation by just checking if this species is present or not to perform the sample clustering as shown on the left. Or you can use the weighted information. So this will include both the species annotation, presence or absence, as well as the abundance for each of the species, as shown on the right. So those two will be both included in our results. So except that, we also provide the statistical studies. So one of them will be the grouping evaluation. As you can check from your bioinformatic information form, so you can define each of your groups, and then we can perform the analysis of similarity by using the anosome analysis. As shown on the right, so those kind of the median data is showing the dissimilarity. So if it is higher, that means we got much higher dissimilarity between groups than the dissimilarity within each group. So that actually indicates we got a very nice grouping when we set up our experiments. Another statistical analysis will be the biomarker identification by using the LifeC. So in this one, again, you define the groups you want to include it in the BI form, and then we will use the different algorithm to identify the biomarkers. So for this part, biomarker will be the different species we identified in the group. So if this bar is kind of getting longer and longer, that means they got much higher significance to represent this group. So as you can see, we got a green group and also red group. So we got different biomarkers for each group. While on the right, the cladogram is actually showing their phylogeny relationship between those biomarkers. So if the Amplicon sequencing can provide all of those information, why do I need the shotgun metagenomics as well? So in this slide, I'm showing you the differences between those two methods. On the right, it's kind of showing you the Amplicon sequencing. So because we will definitely perform the PCR amplification, that actually is more sensitive because we will amplify the signal or each of the 16S ribosome RNA region by using the PCR. So as long as you got that species included in your community, then we can amplify them and detect them. That will also introduce the PCR bias at the same time. So this is kind of the shortage of the Amplicon sequencing. And also, if there is no information about such as the species 2 showing on the second row in the database, then we will fail to detect this information because we don't have that information in our database. The other shortage will be we will fail to detect the novel variations because there will be no variation calling during our amplicon sequencing analysis. Well, on the left, it's kind of showing some information about the shotgun metagenomics. So without PCR amplification, that means low bias, and you don't have to gather information about this target sequence or target region. We can still identify them. And also by having all of those DNA information included in the project, we can perform the gene functional annotation as well. I will introduce the more details about this part in the future slides. So here is the shotgun metagenomics pipeline. And for this one, we will no longer accept the PCR products. So we only accept the genomic DNA. And again, as I circled out, so those will be the specific steps will be included in the metagenomic study. So we need to assemble the metagenome from the data, and then by using the Unigene information, we'll perform the functional annotation in addition to the taxonomy annotation. So Amplicon sequencing will basically include the left part, but by having the Unigene or the gene prediction steps included in this pipeline, we can also perform the functional annotation by using different databases. So here I'm presenting the assembling results from our samples. So in addition to have the assembly results for each of our sample, we also provide unassembled results for the Novomix. So Novomix is coming from the unused reads from each individual samples. So for those reads, we will pick up them again and to perform another round of the assembling. So that will give us the Novomix assembling results. And we will show you the read distribution for each sample and also the Novomix. So this part will be very similar to the Amplicon sequencing. We have the taxonomy annotation by having some similarity or sample clustering and also the biomarker identification. But in the functional annotation part, we will use different database to annotate our genes in our results. So left, I'm showing an example of the annotation from the CAG pathway database. While on the right, we can also perform the sample clustering or group clustering by using the gene annotation information. So in addition to the previous two parts, we also include the resistant gene distribution analysis in our metagenomic pipeline. So here we got two donut figures for two different groups. And within each donut figure, we will include the antibiotic resistant gene distribution in the inner circle and also the predicted gene distribution in the outer circle. And all of those distribution will be annotated at the phylum level. So from those two figures, you can definitely easily to visualize the dominant phylum with annotated ARG, the antibiotic resistant genes, or some other genes, other annotated genes. So this will be very special for the metagenomic sequencing analysis by comparing to the Amplicon sequencing. Well, in the last part, I want just to go through very briefly about the RNA-seq for the Microbial projects. So if you are looking for the single species, then we can provide you the prokaryotic RNA-seq. And in this part, in addition to the quantification, we will also run some special structural analysis for the prokaryotic RNA-seq. And if you want to study the microbial community by using RNA-seq, then we can offer you the Meta-transcriptomics. So in this one, instead of checking what is there, we really want to focus on which species or which gene function is highly or significantly active in my community. So here will be the pipeline for the prokaryotic RNA-seq. As I mentioned, we include both quantification and some gene structure analysis as shown on the right. Well, for the meta-transcriptome analysis pipeline, so left side will be quite similar for the metagenomics sequencing. So we include the gene functional annotation and taxonomic annotation. Well, on the right, we will also include the gene expression analysis and also differential expression analysis to help you find out which genes or which group of the pathways has been significantly activated. So that will be some details for our services. And in the last part, we will go ahead to check a case from Novogene. So this one is for the Amplicon sequencing, but actually from this paper, we included multiple services for the publication. So as you can check from this cute panda, so giant panda is so special to all Chinese and China. So they are actually belongs to the superfamily of the carnivore. However, they feed exclusively on booze. So more than 99% of their food is bamboo. So those bamboo actually are highly enriched in lignin and the cellulose. So how can they digest those kind of inputs in their diet? So in this paper, we are going to answer this kind of question. How does the gut microbiota of giant panda contribute to lignin and cellulose degradation? And if you check their development from their lifetime, so they have to adapt their diet from the breast milk to the bamboo during their growth. So in this study, they collected more than 300 of feces from 12 giant panda, and those panda will be divided into different developmental stages from S1 to S4. And also they included the Amplicon sequencing, the shotgun metagenomic sequencing, and also some de novo assembling and some pan-metagenome studies for this project. However, on the right, I only show you the results for the Amplicon part. So they identify the biomarkers from their gut microbiota, and then they found very different biomarker profile for different stages of the giant panda. As you can see here, so different biomarkers has been showing up at different stages. Well, in conclusion, they found out, so giant panda is actually not well adapted to the degradation of the cellulose and lignin in their highly fibrous bamboo diet. So how they can grow well and get adapted to this kind of diet, because they can maximize their utilization of the energy and the nutrition from the digestible carbohydrates from the bamboo. So they already digested or degraded the bamboo into different carbohydrates, and their body can help them to maximum the intake of their nutrients and those energy. So this is kind of a concentration from Novogene to the study of the giant panda for China. Well, another case study will be from the shotgun metagenomics. As Gurkhan mentioned, so this is a cooperation between Novogene and the University Medical Center Groningen. So in this one, we included 10,000 participants for this study. And here are more details for this. So we will sample, or we will collect the samples, the feces samples from 10,000 participants, and we will run the shotgun metagenomic sequencing by using Illumina platform. And in this part, our objectives included first, to study the microbial components and the functionality. And the second, they want to discover how different factors change the composition and functions of their gut flora. And the last one, they want to see if we can better understand which strain or pathways prevent or cause disease in those 10K participants. So with that, that will be everything for the presentation today. And from now, I'd like to take any questions.
All right, thank you, Wei. Let's start with one question from Mehrdad. What is the use of amplicon metagenomics rather than shotgun metagenomics, while the later one is reliable? Very interesting comment there.
So there will be different reasons when you choose between amplicon sequencing and the shotgun meta genomics, because first, the big reason, so they got different price, and if you have very limited budget, as I mentioned, amplicon sequencing will be a cost-effective way for you to study the microbial community. And the second, as I mentioned in the previous slide, so in the amplicon sequencing, it definitely will introduce some PCR bias during the library preparation, but it also has some advantages, like they can amplify the signal from different species. So once they got the sequence there, we can detect this species. Well, for the shotgun metagenomics, and this one, you definitely will pay more for this project. However, it will perfectly avoid the amplification bias from the process. So it really depends on your budget and also your research purpose. So if you also want to include the gene functional annotation, then metagenomics will definitely a good choice for you.
Okay, thank you. Another question would be, why 97% clustering is considered in the analysis?
So this one is kind of like it's a very so we define the cutoff for the similarity, and this is kind of commonly used for majority of the publications. If you prefer, we can definitely modify the cutoff for you for your special case, but this is a kind of commonly used one. And if you think this is too low, we can definitely level up.
Okay, then we have another one regarding barcoding. That has been answered, but you can maybe comment on it as well. Is it necessary the barcode to be 8 nucleotides? Could it be less or more?
So for our standard pipeline, so we use 8 nucleotide to as the barcodes. But to customize the barcode length, I have to confirm with the lab to see if it is possible to make it shorter or longer.
Right, then we have a question from John. If I have a complete genome sequence which hasn't been published yet, can I provide that to you in order to compare to another sample for resequencing analysis? I think the question is here, if the customer can provide the reference genome.
Yeah, that is quite possible. So usually we'll provide you a list of the references which are already available on our computing clustering. So we will give you some choices, but if you find the listed reference genome is not the one you prefer to use, or you have your own preferred reference genome, you can always send us your reference genome file. However, to run the resequencing, we need two files for your reference genome. First will be definitely the sequence. So that is the assembly file, which is in FASTA format. And the other file we need will be the annotation associated or matched with the assembly file. So that can be in GTF format or GFF format. So that will give us the information about the annotated genes or annotated region for the reference genome. So definitely yes.
Okay, and then there has been a question, which regions are we amplifying for amplicon sequencing?
Which regions? So for different species, we have different regions, such as for bacteria, we use 16S ribosome RNA. So that is a DNA region which encodes 16S ribosome RNA. While for the fungi, we could use either 18S ribosome RNA coding DNA region or ITS coding DNA region. So those will be our standard region or target or market gene for the Amplicon sequencing projects. But if you have your customized region, we can also accept. However, you need to let us know which region you prefer to use or what kind of primers you prefer to use. So we will evaluate those possibilities and then give you some confirmation.
Another question is more general. Will there be any webinars, discussions at any point regarding plant single-cell RNA sequencing services?
Yeah. Well, we already have set up a lot of webinars in previous months. So you can we do have RNA-seq webinar listed on our website, and this is for the eukaryotic RNA-seq, which covers the plant species. And I think in the bioinformatics pipeline webinar, we also included a little bit information about the single-cell RNA-seq as well. But specific to the plant single-cell RNA-seq, we haven't set up a webinar for that topic. But if we got a lot of requests from our potential audience, we can definitely work on that topic and set up one.
Okay, thank you, Wei. Also, a comment on this: all our webinars can be found recorded on our website. Then there is a question about sequencing depth. What is the recommended sequencing depth for a de novo project and a resequencing project?
So it really depends on your research purpose for the resequencing projects. So for that, we usually, if you are looking for the SMP, the single nucleotide polymorphism, so for that one, we may need a little bit lower sequencing depth, at least 10x. While for some structure variations, we do need higher, or copy number variations, we do need higher sequencing depth, such as 20x or even 30x. While for the de novo projects, it depends on what kind of method you prefer or what kind of method you choose or what kind of map you want to get from the data. As I mentioned in one of our slides, if you choose the NGS data, we can only deliver the draft map. So with that, we need at least 100x. While for the complete map for bacteria, we do need long-read sequencing, and we need at least 15x. Well, for the fungus or fungal fun map, we need a combination between NGS data as well as the PacBio data. So I think for PacBio, it will be 17x, but we can, after the webinar, we will share the slides online and we can double check the details for you as well.
Well, then I think we can have time for one more question. The question is, when the DNA sample is extracted from plants, Crow plus 16S amplification can be a problem due to contamination in the amplification process. How can we avoid this or circumvent this?
So yeah, this is kind of common for plant samples. And once we annotate those species by using our database, so the chloroplast samples will be also annotated, and it will be labeled with a mark like this is from chloroplast. And if you need, we can remove those reads after the annotation. So, or the other option is we have some, I think online there will be some kit to remove or to perform the chloroplast exclusive amplification as well. So there will be multiple options, and we can discuss both with you in details based on your condition or situation.
Okay, thank you so much, Wei. I think that's all the time we have today. If there has been some questions that have not been answered, we will come back to you with an email and answer your question there. I'd like to thank you, all of you who attended today to the webinar, and everyone who attended the last webinars as well. All these webinars are recorded, and they will be available on our website, where you can go back and watch these webinars again. Again, thank you very much, and I wish all of you a very good summer, and we will be back in September with new webinars. Thank you and goodbye.